This method can help answer key questions in the postnatal development field about the way milk suckling influences stress signaling in the brain. This technique can be used to capture specific brain areas during the first natural starvation period and to compare them to respective areas shortly after the first natural colostrum feed.
Understanding how suckling influences the physiology of behavior and emotion enables the use of antagonists to various receptors during ex vivo brain tissue analysis.
To obtain the nuclei punches, remove the rat pups by their tail with a gloved hand. After harvesting the brain, rapidly place the whole organ in a polymethyl methacrylate brain mold at room temperature. And immediately use a new razor blade to make 500 micrometer thick slices of the tissue. Laying the slices rostral to caudal in a petri dish as they are obtained to maintain the correct orientation of the sections. When the entire brain has been sectioned, quickly add artificial cerebrospinal fluid without glucose to the dish and incubate the slices for 60 minutes at 28 to 30 degrees Celsius with constant stirring on an orbital shaker. Use a brain atlas and anatomic landmarks on each tissue to identify the brain nuclei to be punched, and place the slice with the nuclei of interest in a new petri dish under a dissecting microscope. Once visualized, use a coring tool to quickly punch out 4 to 6 different nuclei, and rapidly immerse each punched nucleus in 0.06 milliliters of